Hello viewers, hope you all are doing well. Today I am describing the remaining three digestive juices. These are pancreatic juice, bile and Succus entericus. First of all, pancreatic juice. Pancreatic juice is a clear, alkaline type of secretion of the exocrine portion of pancreas containing enzymes that aid in the digestion of all three types of foods, namely protein, carbohydrate and fat. Here is the pancreas. Pancreas is a mixed type of gland because pancreas contains both endocrine portion and exocrine portion. The secretion of endocrine portion, which is hormone, and the secretion of exocrine portion, that is pancreatic juice. Exocrine portion contains two types of cells. One is acinar cell, which secretes enzyme-rich pancreatic secretion, and another one is ductal cell, which secretes bicarbonate-rich pancreatic secretion. Now, come to the composition of pancreatic juice. 98.5% water and 1.5% are solid component. This 1.5% solid component again divided into organic components and inorganic components. Among the organic components, we have carbohydrate splitting enzyme, that is pancreatic alpha amylase, lipolytic enzyme, that is pancreatic lipase, cholesterol esterase and phospholipase A2, and more common name, that is lecithinase. Proteolytic enzymes, trypsin, chymotrypsin, carboxypeptidase, elastase and collagenase. And we have also some nucleases in pancreatic juice like ribonuclease or RNase and deoxyribonuclease or DNase. Now, the inorganic components of pancreatic juice. Among the anion, bicarbonate remain in highest concentration. Except bicarbonate, we have also chloride, sulfate and monohydrogen phosphate in pancreatic juice. And among the cation, we have sodium, potassium, calcium and magnesium. Now come to the functions of pancreatic juice. First of all, neutralizing function. The pancreatic juice is a bicarbonate-rich secretion. That is why pancreatic juice neutralizes the acidity of gastric chyme. Chyme is the partially digested foodstuffs that has just passed out from the stomach. And pancreatic juice also provides a suitable environment for the activation of pancreatic enzymes. Now the second function of pancreatic juice, that is digestive function. Pancreatic juice digests all three types of food due to high concentrations of different enzymes. First of all, digestion of protein. Enteropeptidase or enterokinase and activated trypsin converts inactive proteolytic enzymes into active proteolytic enzymes. Now, how pancreatic proteolytic enzymes become activated, or the process of activation of pancreatic proteolytic enzyme. The proteolytic enzymes, when first secreted by pancreatic cells, they remain as inactive form. They become activated only after they reach the intestinal tract. At first, trypsinogen is activated to active trypsin by an enzyme called enterokinase or enteropeptidase, which is secreted by the intestinal mucosa. Then activated trypsin activates chymotrypsinogen to chymotrypsin, procarboxypeptidase to carboxypeptidase, proelastase to elastase, and procollagenase to active collagenase. Then all the activated proteolytic enzymes convert protein and polypeptides to tripeptides, dipeptides, and free amino acids. Secondly, digestion of carbohydrates. Pancreatic alpha amylase hydrolyzes 50 to 80% of starch, that means both boiled and unboiled starch, into maltose, which is a disaccharide, and maltotriose and alpha dextrins, which are the oligosaccharides, contains 3 to 9 glucose molecules. Digestion of fats. With the help of bile salt, pancreatic lipase converts fat into fatty acid and glycerol. Now come to the digestion of nucleic acids. Deoxyribonuclease or DNase and ribonuclease or RNase are required for the breakdown of DNA and RNA into smaller components. Here are the pancreatic cells with their secretion. Pancreas has two portions, exocrine portions which secretes pancreatic juice and endocrine portion which secretes hormone. We have four types of cell in pancreas. Number 1, alpha cell secretes glucagon. Number 2, beta cell secretes insulin. Number 3, D cell secretes somatostatin, and number 4, that is pancreatic polypeptide cell, secretes pancreatic polypeptide. Now come to the bile. Bile is a dark greenish fluid secreted by liver, stored and concentrated in gallbladder, and released into the second part of duodenum along with pancreatic juice through hepatopancreatic duct or ampulla of Vater. Here is the liver and here is the gallbladder. Bile is secreted from the liver, stored and become concentrated in gallbladder. Right and left hepatic duct together constitute common hepatic duct. Common hepatic duct with cystic duct constitute common bile duct. Common bile duct united with pancreatic duct to form hepatopancreatic duct, and bile is secreted through this hepatopancreatic duct into the second part of duodenum. Now the source of bile, liver bile and gallbladder bile. Liver bile pH is 7.8 to 8.6 and gallbladder bile pH is 7 to 7.4. Here is the composition of bile. Important components of bile are water, bile salts, bile pigments, that is bilirubin, phospholipids, mainly lecithin, fatty acid and cholesterol. Among the liver bile and gallbladder bile, which one is more concentrated? Gallbladder bile is more concentrated because it contains less water. Here are the name of 4 bile salts: sodium taurocolate, potassium taurocolate, sodium glycolate and potassium glycolate. Come to the functions of bile. Bile has no digestive enzyme, but bile is still considered as a digestive juice because bile helps in the digestion of fat and also in the absorption of fat. First of all, digestive function. Bile helps in the fat digestion by emulsification of fat particle by bile salts. What is emulsification? Emulsification is a process by which larger fat globule is broken down into many smaller particles of fat by emulsifying agents. Emulsifying agents of bile. They are bile salts, lecithin, fatty acids and cholesterol. Break up the lipid into many smaller droplets and thereby reduces the surface tension of fat and increases the total surface area of fat for lipolytic enzyme activity, specifically pancreatic lipase enzyme activity, and thereby increases the rate of fat digestion. Emulsification increases the surface area of fat particles by 1000-fold. Now come to the absorptive function. Bile salts of bile form micelle, which vary the end products of fat digestion from intestinal lumen to enterocytes. So bile helps in the absorption of fat and fat soluble vitamins also. Here is the micelle, which is an globular or circular water soluble component. Micelle is formed by the aggregation of bile salts. 20 to 40 molecules of bile salt aggregated together to form a micelle. Here is the structure of bile salt, which have both hydrophilic portion and hydrophobic portion. During formation of micelle, hydrophilic portions of bile salt faces outwards and hydrophobic portion of bile salt faces inwards, and in this way they make the micelle is a water soluble component. We know fat is an water insoluble substance but soluble in nonpolar solvent like ether benzene and chloroform. For this reason, the digestive end products of fat are also water insoluble. These water insoluble digestive end products of fat enter into micelle. Micelle carries them or transport them through the watery intestinal medium to the enterocyte. After coming into the enterocyte, these water insoluble digestive end products of fat spontaneously diffuse out to enterocyte, and micelle again return back to intestinal lumen until all the digestive end products of fat become transported. In the presence or an abundance of micelle, about 97% of the fat is absorbed. In the absence of the micelle, only 40 to 50% can be absorbed. Now the third function of bile, that is excretory function. Bile is a vehicle for excretion of bilirubin, steroids, barbiturates, excess cholesterol, alkaline phosphatase, and many metabolites and drugs. And lastly, bile salts of bile also increases the bile flow. Now come to the last digestive juice, that is succus entericus or intestinal secretion. Succus entericus is the intestinal juice secreted by the glands in the wall of the duodenum. Come to the composition of succus entericus: 98.5% water and 1.5% solid. Among the solid components, we have organic components and inorganic components. Carbohydrate splitting enzyme like maltase, isomaltase, sucrase, and lactase breaks down maltose, isomaltose, sucrose, and lactose into absorbable monosaccharides. Proteolytic enzyme, several peptidases like aminopeptidase, dipeptidase, tripeptidase, carboxypeptidase for splitting small peptides into free amino acids. We have an activator enzyme here also, that is enteropeptidase or enterokinase, which is secreted from the small intestinal mucosa for the activation of the inactive proteolytic enzymes. Small amounts of intestinal lipase present also here for the splitting of neutral fat into glycerol and fatty acid. Among the inorganic components, we have some cations like sodium, potassium, calcium, and magnesium, and we have also some anions like bicarbonate, chloride, monohydrogen phosphate, and sulfate.